All right. Hey there. Welcome to this excellent meeting to talk about the biology program at The Ohio State University at Marion. And a little bit about The Ohio State University at Marion. So Ohio State University is a single university. So on any campus, if you get a degree, you get an OSU degree. You also have access to lots of the resources that are centralized, like libraries and basically faculty and other things like that. So we are basically one university. And at the Ohio State University at Marion, we started offering the biology major in 2015. And so the program has really taken off since then. And in fact, we were able to build a new science and engineering building in 2018, which you would be able to take your classes in. So this is a picture of our new building here. So nice building, lots of windows, very beautiful, beautiful lab space with very like kind of flexible seating. We have classrooms of different sizes with, nice projection systems and lots of common areas where people can hang out. So good building. All right, so I was going to ask all of you what career paths you're interested in. So go ahead and in the little chat there, just tell me what kind of career paths you are interested in pursuing. All right, let's see here. What do we see? We see, all right, somebody's interested in potential career in medical school. Yeah, all right. I see that dental school. Yep, nursing potentially. Yeah, interested in research. All right, radiology. All right. Yep, yep, lots of diversity. A lot of things you can pursue with a biology degree. Okay. Let me close this. Okay. So there are lots of potential career paths you can take with a degree in biology from OSU Marion. Lots of biomedical career paths where you're doing care or you're potentially doing research to answer biomedical questions. Many people who have a degree in biology go on to work at a college or a university as a professor or an instructor. You may be doing primarily research, or you might do primarily teaching as your job. You can use a career in biology to kind of further your career in agriculture. So many people are involved in management and feeding the world and in trying to reduce disease and improve yield. A lot of people do research in agriculture. So those are other possibilities. There are careers in conservation. I have many students who are interested in that as well. Going out and surveying what is out there in the world, identifying it and writing up what you see, doing research in terms of the most efficient and effective conservation techniques, and also outreach to explain to people why conservation is important. Government and private research institutions hire people to do all sorts of different things, including research and education. And I have a lot of students interested in zoos, aquariums, and museums as a possible career path. And so a biology degree can prepare you to go off and learn how to do animal care and education and also research as well. All right, so at OSU Marion, these are our general offerings for the biology major. So we have the pre-health professionals specialization of the biology major. And so we recommend you start with the kind of two-part series of introductory biology. So in 1113, you learn about, you learn about cell structure, you learn about DNA, you learn about all sorts of related things like that. In 1114, you learn about evolution, you learn about biodiversity, you learn a little bit about anatomy and physiology of animals and plants, and you learn about ecology. In 2510, you study human anatomy and kind of get to learn about all of the basic systems in the human body. Physiology, you get to see how these systems work. Microanatomy, you get to look at the what you can learn from looking at cellular anatomical features. Let's see, if you need to take Bio 3401 or integrated biology as a required course, and that's a course that integrates the material from Biology 1113 and 1114, and the OSU Marion campus. We offer the topic for that class as being applications of biotechnology. Evolution, so you get to learn about microevolution and macroevolution, and how evolution is applied to all sorts of things, like medicine and health as well. Cellular and developmental biology, so you get to learn more details about the cellular mechanisms in biological systems, as well as developmental pathways. Comparative vertebrate anatomy, you get to have a little bit of a deeper dive into more diverse kinds of vertebrates, and thinking about how some features they have in common and some features are different. Molecular, or MolGen 4500 is general genetics, and so you get to learn a much more intensive view of the principles of genetics. Microbiology, basically you get to learn about the microscopic world of living things. And if you are here because you are a nursing student, you have the kind of pathway below. So you'd start with Biology 1101, which compresses the material that you get in 1113 and 1114 into a one semester class. And then anatomy, physiology, and then microbiology. All right, so the faculty at OSU Marion are all experts in their particular field. So I am Susan Gershman, and I'm in the Department of Evolution, Ecology, and Organismal Biology on the main campus in Columbus. And I study the evolution of animal behavior. Dr. Ruben Petreka studies, is in the Department of Molecular Genetics, and he studies cancer and DNA damage repair. Dr. Jonathan Khaled is in the Department of Evolution, Ecology, and Organismal Biology, and he studies the ecology and evolution of fossil mammals. Dr. Frances Sivakov is in the Department of Evolution and Ecology and Organismal Biology, and she studies the ecology of insects. And Dr. Melissa Petreka is a senior lecturer at OSU Marion, and her specialty is the regulation of immune system inflammatory cells. And so some things that we can offer on the Marion campus is we have the opportunity for students to do research in the labs of these various biologists and microbiologists. We have other resources as well, like for example, we have a prairie that was reconstructed, I don't know the exact year, but looks like the 70s from this picture. And so we have the opportunity to do lots of research projects out on the prairie as well. All right, are there any questions so far? Anyone want to know anything? You can either ask your question aloud or type your question in the chat if you want to. Yeah, the nursing track is, I had a question here, is the nursing track pre-nursing? Yes, it is pre-nursing, so it prepares you to go to nursing school as well. I got opportunities for research. So we have a couple labs that are doing different kinds of research. So we have a molecular genetics lab. Dr. Petreka has students doing work, some lab work, some computational work. Dr. Khaled is having students do anatomical work, measuring characteristics of fossils to learn things about their ecology and evolution. There's also my lab, Susan Gershman. I do research on the evolution of animal behavior, and so we have students doing projects right now on crickets, looking at cricket behavior, and I think Frances Sivakov as well. Dr. Sivakov has potentially places for students to help do some of her prairie projects with her, or some of the work analyzing seeds that she is collecting in the field. And yeah, we also have researchers in chemistry as well. So I had a question: can I complete all the requirements of the degree at Marion, or do I have to finish in Columbus? You don't have to go to Columbus for anything. If you want to take a class that they offer, I think you can take up to 3 classes from the Columbus campus and have that not cost anything extra. You just pay the Marion fees for those. So that's a nice feature as well. But we do have all of the classes. But if they have something that you are really extra interested in and you want to go do, you can. All right, so I'm going to keep going. Oh, cost differences with Columbus, yes. We actually charge less per credit hour than Columbus campus. And so if you take, I think, up to 3 classes on Columbus campus, you get charged the Marion rate for them. So that's kind of a nice feature as well. What's the relationship between faculty members and students? Are you all accessible? Yes, we're all just sitting there with our office doors open. People come in all the time just to chat with people. The classes are relatively small, especially as compared to Columbus campus. We tend to have 24 or fewer students in each class. So you get to know your professors. I've had some students 2 or 3 times, and so I really get to know them quite well. Do we have teaching assistants in lab? No, actually, that's another bonus that on main campus, you will not necessarily get to interact with your professor because you will have most of your interactions with teaching assistants who are usually grad students or instructors. But on Marion campus, all of the labs are taught by the faculty and full-time lecturers. And so you get to have lots of good interactions with those people. Great. So I have a short, presentation that's kind of a summary of material that I'm kind of working on for a lecture, but I thought y'all might be interested in this. So I'm going to talk a little bit about vaccines, but feel free to ask me questions or whatever as I'm going along. All right, so in order to talk about vaccines, we have to talk a little bit about immunity, right? So immunity is defined as resistance to or protection against a disease-causing pathogen. And there's 2 basic elements to immunity. The first is innate immunity. And so this can include all the things that are the first line of defense, like your skin, that stops, pathogens or bad things from coming into your body, mucus, oils, you have substances in your saliva. And actually, if anyone is interested in this kind of innate immunity or inflammatory response, Dr. Melissa Petreka is an expert on this. So you can go ask her questions. The other component of immunity is adaptive immunity, and this is the part of immunity that deals with the interactions that happen between antibodies and antigens, which I'll talk more about in a second. All right, so I'm going to use the word pathogen quite a bit. And by pathogen, I mean anything that is harmful to our bodies. So it could be a bacteria, a virus, could be a protist or something like that. So it could be something like influenza B, right? I'm also going to use a word that sounds like pathogen, which is something different. So on the outside of a virus like influenza B, there's these little nubbins, okay? Those little nubbins are called antigens. So antigens are these kind of protuberances that come out of the outside of many viruses and potentially bacteria and other things as well. And so if you look at them up close, you can see they have all sorts of little nooks and crannies, right? And each of these little nooks and crannies has the potential to stick to other molecules. So I am going to, because of these trying times, talk a little bit about SARS-CoV-2, and I'll talk about vaccinations with respect to SARS-CoV-2, because that's on everybody's mind. So SARS-CoV-2 is the name of the actual virus, the novel coronavirus, and the disease that SARS-CoV-2 is called the disease is COVID-19. So you can see in these nice pictures of SARS-CoV-2 that these are the little antigens sticking around the outside. And people who have done a more detailed analysis of their structure would show you this picture. So you can see this is a very complicated little structure with all sorts of little nooks and crannies that might potentially bind to other molecules. All right, so the proteins or the antigens on the surface of invading cells have the potential to attract the attention of the immune system. And so this Y-shaped molecule here, this antibody, is the immune system molecule that is the one that comes over and binds to an antigen. So antigens bind to, or antibodies come and bind to antigens. Antibodies are made by B cells. So B cells will generate billions of different antibodies, and antibodies differ in the little tips on the ends of each of their arms that will fit antigens differently. And if you actually want to know what an antibody really looks like, it looks like this. So it is actually also a very complicated, nubbly looking molecule as well. All right. So most antibodies are secreted into the lymphatic or the lymph or the lymphatic system of the body, but some of the antibodies remain bound to B cells. And so these particular antibodies are referred to as B cell receptors. Okay. All right, so let's talk a little bit about adaptive immunity before we get much further. So I mentioned that adaptive immunity is a part of the immune system that has to do with antibodies, right? And so I'm going to show you this kind of description of how adaptive immunity works. It's very simplified, and it's shown with respect to the SARS-CoV-2 virus. So here's a picture of the SARS-CoV-2 virus. And so it's got an outer casing with its little nubbin antigens around the outside. And then inside it's got a little squiggle of RNA, the genetic material. So basically, when somebody gets infected, the virus enters the body, and inside the body, it will replicate inside of the cells. And so eventually the virus ends up getting eaten by an antigen presenting cell. And so the antigen presenting cell is named that because after it basically digests the SARS-CoV-2 molecule, it'll take one of the antigens off the outside and kind of stick it on the outside of the cell. And so that is why we're calling this an antigen presenting cell. It's basically like saying, hey, look what I found, and sticking it on the outside of the cell. All right, so once the antigen presenting cell presents the antigen, a helper T cell will come over and recognize the shape of that little nobbin, and that'll help recruit other immune cells to come over and help. And so some of the other immune cells that are coming over are like B cells and cytotoxic T cells. And so this triggers the two kind of arms of adaptive immunity. So the B cells will participate in antibody-mediated immunity, and the B cells will help make the specific antibodies that, like the little nooks and crannies on these antibodies, will exactly fit the nooks and crannies on the antigens. And so then these antibodies will come over and bind to the antigens of SARS-CoV-2. And that will basically help the body of the host to be able to find the pathogen and destroy it. So it tags the pathogen. And so the body recognizes that there's some kind of a molecule that's covered in antibodies, and the body needs to come over and basically break that down. The cytotoxic T cells participate in cell-mediated immunity, and so that basically calls over cells to destroy the host cells that happen to have virus inside of them. One really important thing that we have to know is that some of the B and T cells are going to stay in the body and help the body recognize if the body gets exposed to the same virus again. So let's talk a little bit about how that happens. So these are B cell receptors, so they each have an antibody on the top of them. And the antibodies are all different. There's all sorts of different antibodies with all sorts of different little tips on the ends of them, just waiting to, match up with the nooks and the crannies of some antigen that enters the body. Okay, so when you have a B cell receptor bind to an antigen, that triggers that B cell to rapidly reproduce and reproduce. And B cells have this unique quality that allows them to mutate at a much higher rate than other cells would ordinarily do. So they're mutating in all different places all over the genome of those B cells. And that causes the antibodies on the outside to have all sorts of little like nubbins and nooks and crannies on the end of them that could potentially match up with all different kinds of antigens. Okay, and so when you find a receptor that happens to bind to an antigen, that will trigger that particular lineage of B cell to reproduce much more and outcompete the other B cells. And so you end up with a whole giant pile of these B cells and these antibodies that happen to bind to this antigen. And so this is how you refine the bond between the receptors and the antigens so that you end up with like the best possible antibodies that bind to the antigens as well as they possibly can. So some of those B cells will become memory cells and they'll be kept aside. And so they are just sitting there lying in wait. And if the host happens to encounter that same antibody or that same pathogen again with antigen, that same antigen on the outside of it, then the host can mount an immune response very quickly. So I just point out that you can actually test for the presence of memory cells in a person. So there's a test called the ELISA antibody test, which is the enzyme-linked immunosorbent assay. And you start out with kind of a plate like this. And inside of these little wells here, they've taken a bunch of SARS-CoV-2 antigens and just glued them to the inside of the wells. And you can add, well, actually in this case, I don't, you do blood, maybe, I don't know if it's blood or spit test, but whatever. So you can add some of the patient's, I guess, blood usually. And so then if the patient happens to have the antibodies that bind to the antigen, they'll bind. If they don't happen to have those antibodies, then they won't bind. So then you kind of wash the plate out and wash all the excess fluid out of there. And you add something that glows if the antibodies are stuck to the antigens. And so only the wells that have antibodies that happen to stick to these antigens will glow. And so that tells you if this person happens to have the ability to very quickly make the antibodies for a particular antigen. All right, so what is a vaccine anyway? Well, a vaccine is something that stimulates the production of antibodies for a specific disease. So if you become infected, then you have some kind of an immune response. So that's your primary immune response. And at the end of that primary immune response, you'll start to produce memory cells, which will get stored. And so then if you become infected with that same pathogen again, you already have those memory cells, and that allows your body to very, very quickly ramp up and produce a really large immune response so that you're able to basically defeat that pathogen. You might not even feel sick at all that time. You might not even know you were infected. So that is basically what a vaccine does. The vaccine is something that allows you to make memory cells so that you you do not become sick the next time you're exposed to that antigen. I also wanted to talk a little bit about active versus passive immunity before we get on to vaccinations. So active immunity is when you have resistance developed in response to some kind of a stimulation by an antigen. So it could be that you actually got sick with something or you were vaccinated. And usually, and always actually, active immunity is characterized by the fact that you produce antibodies. Passive immunity is immunity that you get by being given antibodies by somebody else, basically. And you can get these naturally or artificially. And so I'm going to talk about this for a second. So with active immunity, so two kinds of immunity, active and passive, as I said. With active immunity, you can become actively immune after you become sick. So you could get infected and then, be really sick and then fight it off. And now you have active immunity. Sometimes you can get active immunity without actually having symptoms. So you could be asymptomatic, but your body has been exposed and developed memory cells. And you could have active immunity after vaccination. So these two mechanisms are natural and this is acquired, right? With passive immunity, you could acquire antibodies as a developing fetus through through the placenta, right, from your mom. You could get passive immunity as an infant by drinking milk. So some antibodies are passed through milk. So these are two natural ways to have passive immunity. And you can also get passive immunity in an acquired way because you can actually receive antibodies or antiserum from another source, from another host, and then you have passive immunity. All right, has anyone ever heard of antibody therapy before? Oops, it advanced it without me meaning to. Yeah. All right, so what do you think? No, you haven't heard of it before? Okay, has anyone heard of it? Okay. Have you heard about it in the news maybe? Yeah? They're the antibody treatments that Trump got. Yeah, that is true. He got antibody treatments, right? That might be something that you might get at the hospital if you come in with kind of serious symptoms of COVID-19, right? So let's talk about how that works. Okay. So there are antibody therapies for not just COVID-19, but for a couple of other conditions as well. So it's possible to treat an infected patient by injecting them with antibodies that happen to match the pathogen that they're infected with. And that can be really helpful for the patient because it can stimulate the patient's own immune system to attack the pathogen. And so even though the patient's immune system maybe wasn't able to find the pathogen on its own, once you give the patient all of these antibodies, the antibodies will kind of glom all over that particular pathogen and help the patient's body to defeat it. And so antibody therapies can be effective against pathogens that are capable of evading the unstimulated immune system of the patient, but are susceptible to the stimulated immune system. So if the person's very, very sick, giving them lots and lots of antibodies may not actually help if their own immune system can't rise to the challenge. But if they're moderately sick, this can kind of give their immune system a bit of a head start. So let's see, I have a question. So the antibodies can be made in a couple of different ways. You can have a healthy donor, so somebody who's been infected with the pathogen and then they recover, like, I don't know, Tom Hanks, right? So people were donating blood. People who had been recently infected with COVID-19 were donating blood, and then people could get if you were moderately to severely ill with COVID-19, oops, it says patent, but patient. If you are moderately or to severely ill with COVID-19, you might receive a blood node donation from somebody who recently had COVID-19, and that would give you their antibodies. Or the antibodies that happen to match the pathogen can be isolated in the lab and copied. I don't have time to get into that, how you do it too much, but we covered that in one of the classes I teach. But and so this is called monoclonal antibody for treatment of COVID-19 is the official name of this particular treatment. There are two different commercial products. One of them is a combination of two drugs, which I cannot pronounce. The other one is this drug here. And so this is basically a drug that just contains antibodies that happen to match COVID-19, that happen to match SARS-CoV-2. So can anybody tell me, yeah, every time I go to click the chat feature, it advances the slide, but does anyone happen to know why these end in MAB? Any ideas for that? Oh, got some ideas. Yeah, AB is for antibody. Yep. And you got, the M is for monoclonal. Yeah, you got it. So this actually can be kind of useful if you're just watching television and they have ads. If you see some drug that they're selling for some other purpose and it ends in MAB, it means that it's a monoclonal antibody medication. So it's basically a bunch of antibodies that have been produced in a laboratory that happen to match one specific antigen. And so the goal behind this kind of monoclonal antibody treatment is that it might be able to keep people who are vulnerable and are infected with SARS-CoV-2 from requiring hospitalization. So it's possible that if somebody goes into the hospital, that they maybe could be treated as an outpatient after they get one of these series of medications, and it won't get serious enough that they actually have to be hospitalized. Okay, so let's just compare and contrast some of the similarities and differences between the two things. So antibody therapy, you are treating a person who is infected, okay? So you would go into the hospital because you're already infected to get antibodies. Vaccination is something you give to a healthy person to avoid getting infected. Antibody therapy gives you passive immunity. Vaccinations give you active immunity. So antibody therapy, you don't produce any memory cells at the end. So you get the person's antibodies, but it will not, you don't have memory cells, so you don't have a long-term effect. Vaccination, you get memory cells, right? So the upshot of this is for antibody therapies, you get an immediate beneficial effect of being able to get all these antibodies in your body, but it doesn't last for very long. So it's a very short-term thing, but it might help you long enough so that your immune system can take over and you can get better. Vaccination, it takes a little bit longer to start working, but it will work for much longer periods of time. All right, well, I don't think we have time to do this today, but I was going to invite you to do an exercise in which you got to kind of go to this link here and try and figure out where all of these different where all of the different things that we talked about fit into this kind of matrix of being natural versus artificial and active versus passive. But I'll let you just think about that on your own. All right, now I'm going to kind of review some of the types of vaccines, and there are more vaccines than I am presenting, but I'm just going to present the types of vaccines that are under consideration for treatment or for preventing infection by COVID-19 or SARS-CoV-2, the cause of COVID-19. So the basic types are whole pathogens, either inactivated or killed vaccines, or live attenuated, meaning weakened vaccines, pieces of pathogens, which I'll talk about in a minute, or methods that use a virus to carry antigen genes, or what they're called naked antigen genes, in which the DNA is put directly into DNA or RNA is put directly into a person. So let's look at some of these. All right, so an inactivated virus vaccine is basically the RNA. If this is a picture of SARS-CoV-2, and the RNA has been destroyed. Okay, so the genetic material is destroyed, and so it's basically just kind of a hull, right, with no DNA or no RNA on the inside that works. So this viral particle isn't able to replicate inside the body. Okay, so you actually have to inject quite a bit of this into a person. Because it's not going to reproduce once it gets inside the host. And so you need lots of doses, but basically it works by, eventually one of these antigen presenting cells will engulf it and then present the antigen that was on the outside here. I should point out, because this doesn't replicate, we need to use something called an adjuvant, which is a substance that's used to strengthen the immune response. So that's what these little speckles are showing. Because without the adjuvant, your body wouldn't have a strong enough immune response to know to come over here and do something about this little dead virus that you have in your body. All right, and so then the antigen presenting cell presents the antigen to a helper T cell, which stimulates the antibody mediated immune response, and you produce all of your little antibodies, okay? And then you make memory cells. The live attenuated virus vaccine works by taking a SARS-CoV-2 virus and weakening it. So they basically were able to go in there and alter the RNA so that it is weakened. And then that gets picked up by an antigen presenting cell. Because it can still replicate, even if it doesn't have the ability to do some of the things that make you really sick. You don't need to add an adjuvant, right? It's able to reproduce and make more copies of its little weak self. It gets picked up by antigen presenting cells. They present the antigen to the helper T cells, and it's able to create an immune response as well as a memory. So compare and contrast the two, it is actually much easier to store a dead thing than it is to store an alive thing, right? People sometimes get worried that if you have something that's alive, it will mutate to become harmful. That isn't a risk with inactivated virus vaccines, but it is weaker and requires an adjuvant. You get a really good, strong immune response to live attenuated virus vaccines. They're a little harder to store because you have to keep them alive, and people do worry about a risk of mutation, even though statistically this is very unlikely. All right, the next kind of vaccine are a subunit vaccine. And so what they do is they don't put the whole virus particle into a person. They just take the little nubbins, the antigens, plus an adjuvant, and put that into the person. So again, it's picked up by an antigen presenting cell. So the antigen presenting cell is just squishing around in there and it finds these antigens. It's like, hey, look what I got, it says to the helper T cell. And then that's used to stimulate the antibody-mediated immune response to make antibodies. It's very safe because you can't get sick by a bunch of nubbins being put into your body, for sure. But it's much weaker and it requires both an adjuvant and usually several doses. There are actually other ways to kind of goose these to make them a little bit less weak, but I don't think they're under consideration for SARS-CoV-2, so I'm not going to talk about them today. All right, so I know it may have been a while since you've thought of this, but what is transcription and what is translation? Does anyone remember? You can either say it aloud or put it in the chat. All right. Yeah, we get like copying stuff. Yeah, transcription is like copying stuff, right? So yeah, that's good. The thing with the ribosomes. Yeah, you got with the translation, you got the thing with the ribosomes. Yep. Does anyone remember what the main point of transcription and translation are? How you make DNA? Well, kind of. Yeah, no RNA. Yeah, it's how you take the DNA and you make it into RNA to make it into proteins. Yeah, cool. All right, let me show you a picture of that. Okay. All right, so this is the simple picture. So DNA gets transcribed into a molecule called RNA, which becomes messenger RNA. So the information in the DNA is used to make a molecule of RNA, and then the RNA leaves the nucleus and gets made into the information in the messenger RNA is used to make a protein. Okay. This is the more detailed picture. If you do happen to remember some of this, I kind of like this picture because it looks like a party with confetti. Although I don't believe these are confetti, I believe these are amino acids. And it's just showing the same thing, that you start out with your double-stranded DNA, and that information is being used to make RNA. That RNA is processed to make messenger RNA, which is sent out of the nucleus like this. And then this messenger RNA out of the nucleus is read by this ribosome, and the ribosomes bring over the amino acids to make a chain of amino acids, and so a chain of amino acids is a protein, right? So we're basically making proteins. Okay, well, the reason I brought all that up is to talk about viral vector vaccines. So there's two kinds, basically. There's a replicating viral vector vaccine. And so this method uses some kind of a non-coronavirus vector. And when we say vector in biology, well, in math, it's something, it has what is magnitude and direction, right? In biology, it just means something that's carrying something somewhere else, okay? So in this case, it's some kind of a virus that's going to carry the information that we need. So what we do is we put this other virus that we know works pretty well for making vaccines out of, and we take a little bit of its RNA and we stick in there the gene for the antigen of SARS-CoV-2. So this little gene basically has instructions to make SARS-CoV-2 nobbins, not these nobbins like this or those nobbins like this. No, make the nobbins that go on the outside of SARS-CoV-2. Okay. So the virus will go replicate inside of the body. This other virus, this virus isn't SARS-CoV-2, it's another virus. And then the antigen presenting cell will pick up this other virus, and inside of that cell, it'll produce the antigen because it has this little bit of DNA or RNA, in this case RNA, that says, make that antigen. And so the cell makes that antigen, and there it is. It presents it on the outside, where it gets the attention of a helper T cell, which activates immune response and memory. There's also viral vectors that are non-replicating. And so you also will take a little bit of its DNA or RNA and substitute for the DNA or RNA that you're interested in. So in this case, we've taken the RNA and substituted in the gene that encodes for the nobbin that goes on the outside of SARS-CoV-2. So this is an antigen gene, okay? And it works the same way, except that this, this viral vector is not capable of reproducing itself. So you have to put a lot more of it in to get injected. You have to get injected with a lot more of it for it to be effective, but it works the same way. Basically, an antigen presenting cell will pick it up, and then the virus, like all viruses do, makes its host, replicate itself and produce its protein products. And when it does that, it makes these little antigens. There we go. It's making the antigens that go with SARS-CoV-2, and then the antigen presenting cell presents those, etc. So comparing and contrasting these two things, you get a really nice strong immune response from a replicating viral vector. One weird thing is the host may actually, if you have had exposure to the vector before, you may actually already have a way of dealing with it. And you might actually inactivate the vector, which is kind of a weird, cool trick the human body can do. If you have the non-replicating vector vaccine, it has the advantage of being able to hold larger genes. So if you happen to have some pathogen that has like a really big complicated nobbin or multiple nobbins on the outside that you want to make, you can put it in there, but you need more of it. Oh, let's see, we have some questions. Hold on. Trying to get it to give me my chat box back. All right, well, it's not letting me do it. Okay. There we go. Yeah, so I have a question. Most of us already have immune memory for measles and adenovirus. Does this affect the immune response of these modified versions? It could have if they picked, like this is one reason why they probably don't use measles as a vector. They do use some kinds of adenovirus, right? But I think that they attempt to find ones that we don't necessarily have an immune response to. But yeah, it's technically possible that you could potentially have already like recognized that adenovirus, and so when it gets into your body in the vaccine form, you're like, nope, killing it, and then you have to do something else, I guess. Okay, here's something new you might not be aware of. This is called virus-like particle vaccines, and so it's called a virus-like particle. And it mimics a real virus, but it has no genetic material in it. So basically we just make this thing. So we make something that has antigens on the outside and it's just hollow on the inside. And again, it gets engulfed by an antigen presenting cell, which presents the antigens and stimulates the immune system. These are very safe because they're not actually real living things. Well, viruses aren't alive anyway, but they're not, they don't have the potential to spread or to reproduce or anything. They're very easy to store. They're more technically difficult to make, and so they can be more expensive and time consuming to make. And they are weaker, so it takes several doses to be able to get an immune reaction to one of those. And then finally, we have DNA and RNA vaccines. So DNA vaccines are bacterial derived from bacterial derived DNA plasmids. So a plasmid is like a little circle of DNA that's contained in bacteria. And the little circle of DNA has been modified to insert this little bit in here. And this is the gene that codes for the SARS-CoV-2 antigen. So this is the instructions for making the SARS-CoV-2 antigen, okay? And so what you can do is take this little DNA plasmid and just kind of put it into a person, and then this will get taken up by the person's cells. And it doesn't go into the nucleus of the cell, so it's not like messing with the major functions of the cell, but it is, well, let's see. I think this picture is going into the nucleus. I do not think it really goes into the nucleus though. It gets, yeah, let's see. It gets transcribed and translated to make the antigen. I'm sitting here thinking about this here. I should have thought about this before I started presenting this, but I guess it could get picked up by the nucleus of the cell. I think it could also potentially replicate outside of the nucleus of the cell, though, as well. So either way, it is going to make more copies of itself, and then that little section of the gene is potentially going to code for antigens. And so when proteins are being made, the proteins will include proteins to make these little antigens on the outside. Let's see here. You have a comment? Yeah, we have that the RNA polymerases are in the nucleus. Yeah, that is true. Yeah, I mean, this is kind of a new technique. There are actually very few DNA vaccines that are currently, actually, I don't know of any that are approved right now, so I'm a little bit less familiar with this. Yeah, you would need the RNA polymerases from the nucleus, so I suppose it would have to go into the nucleus to be made. Let's see. All right, so again, the antigen gets recognized by the antigen presenting cell, and then that stimulates helper T cells, and then you get your immune response. So RNA vaccines are able to replicate actually in a way that doesn't go into the nucleus of the cell. So they are put inside of like a little kind of bubble of lipids that act a little bit like a cell membrane around the outside, or a vesicle just to hold on to it. And then inside there, you have a little bit of messenger RNA, and that's the little bit of genetic information that codes for how to make an antigen. And so that is injected, and then it gets inside of cells. And so that is already in RNA form, so it would not need to be in the nucleus to replicate itself. And so it's able to self-replicate. And then also these little bits of RNA are read and made into proteins. And so the proteins that are made are these little antigens. So then it produces antigens. It causes the cell to produce antigens as a product. And those are picked up by antigen presenting cells and presented, and they stimulate the immune system. So one nice thing about the DNA vaccines is they're very cheap to make, in that they require fewer steps than some of the other kinds of vaccines, and they're easy to store. Another nice thing is that they also stimulate the cell-mediated response, as well as the other immune pathway. For RNA vaccines, they're cheap-ish. They're a little bit more expensive and hard to make than DNA vaccines. They are also harder to store because RNA is a little bit more fragile. But one nice bonus is that you actually require a smaller dose of them than DNA. So that's kind of nice. All right, so let's talk a little bit about vaccine testing. So once a vaccine is kind of invented, you have to test it, make sure it works. And we hear a lot about vaccine testing, but I just wanted to explain what that was a little bit. So we hear about 3 phases of vaccine testing. So in phase 1, this is when this experimental vaccine is given to a small group of healthy adult volunteers. And this is a step that's done just to assess whether there's health risks. So they monitor these people. So some of them will be given the vaccine, and some will be given a control like a placebo of some sort. And then all of these people are monitored to determine whether there's health risks. And I know that if you've been following the news, there were a couple of vaccines in which one of the volunteers became sick, and then they would have to stop the study and figure out, did they get sick because random reasons, or did they get sick because of the vaccine? And so far, none of the vaccines have actually caused anyone to get sick, but this is the purpose of phase 1 studies. Phase 2 expands the pool of people who are included, so it can include older adults and children. And this is the phase in which you determine the immune response to the vaccine in people of different ages. And you determine the immune response using kind of a ELISA, or an antibody test, to determine how strong an immune response the vaccine causes in the people. And so this would be a time where you'd say, oh, maybe this vaccine is not strong enough. It doesn't cause enough people to make memory cells. Maybe we need to add an adjuvant to it or something else to make a better immune response, right? Or maybe it's not effective in certain groups of people. Maybe it's not effective in children or older adults or somebody like that. So this is the part in which they have to, kind of troubleshoot in a more diverse group of people. And then in phase 3, this is when they have to get really diverse. And so this is the point at which you want to pick people, people of all races, people in lots of different locations in other countries, people who are genetically dissimilar from one another. And you're still looking to see things like health risks and immune response, right? But at this stage, you're also looking at the effectiveness of the vaccine. So you're including lots and lots and lots of people in this study. And these are people who are going to get exposed to other people who have SARS-CoV-2 or whatever the disease is. And so what you want to do is compare how many of so for phase 3, you're injecting people with the vaccine and injecting people with a placebo. And you want to compare how effective the vaccine is at actually preventing people from getting SARS-CoV-2 and contracting COVID-19. Okay. I've heard a lot of people talk about being worried about SARS-CoV-2 vaccines because of how quickly they were rushed out. And I just wanted to draw people's attention to something you might not be aware of, but every year there are large numbers of people who are generating influenza vaccines. Every single year they have to generate new influenza vaccines, right? So they have to get it from the point where they don't even know which influenza strain they're going to put in the vaccine, all the way up to distributing it. And that's like every single year, right? And that's harder than just coming up with a vaccine for a single virus that we have identified already and we know a lot about. So for example, for the flu vaccine, they have to go out and sample all of the flu that's out there at the moment. They have to kind of use techniques that we'll learn about in some of the classes, phylogenetic techniques, to assess which lineages are likely to be the ones that are going to be a problem a year later. They have to pick, they usually pick, I think, 7 of them, 7 different strains or around 7. So they have 7 different strains involved in there. They have to figure out how to make an effective vaccine for those 7 different strains. They have to manufacture it, license it, package and distribute it, and then vaccinate the people. So this happens every single year. Although things are moving very, very quickly with SARS-CoV-2 vaccines, I would not be concerned about the fact that these are rushed. This is a tight schedule, but it's something that people are familiar with dealing with. All right, we're almost at the end here. Has anyone heard of herd immunity before? Anyone know what it means? Yeah, you've heard of it? Oh, you've heard of it. Haha. All right, so what is herd immunity? Who can have herd immunity? Something about cows? I mean, I bet cows have herd immunity. Yeah, when enough people are immune, then you can protect folks who can't get vaccines. Yeah, that's it. Basically, it's allowing people who are vulnerable and unable to get vaccinated to be protected by everyone else getting vaccinated. So basically it's defined as when a large percentage of the population has become immune to an infection, there's an indirect protection for the remaining people. So these could be elderly people, people who have immune problems that prevent them from receiving vaccinations, small children who haven't been vaccinated yet, right? And so there's lots of simulations out there. This isn't actually a real simulation, it's just a GIF of a simulation, which is why the same thing is happening each time. This is basically just showing that if the blue dots are people who are unvaccinated, maybe because they're unable to be, and the yellow dots are the people who are vaccinated, every time the line will start randomly somewhere in the middle, and every time the line goes to somebody who's vaccinated, it basically stops. Every time it goes to someone who's unvaccinated, it spreads. So the more people that you have who are unvaccinated, the more likely it is to spread and the more people who become ill. And then finally, I just wanted to point out that there are great resources available if you like to follow the news and you're interested in vaccines. Vaccine Tracker is a good site. It kind of updates fairly regularly, telling you what the status is of all of the different vaccines. I just threw these up here. These are all the vaccines that have been approved somewhere. Only the 2 mRNA-based vaccines are approved in the US. but lots of other countries have approved all sorts of other vaccines. They've approved inactivated vaccines, replicating viral vector vaccines, non-replicating viral vector vaccines, subunit vaccines. There are others. There's so many more vaccines that are in all different phases of testing. Some of these are DNA vaccines, some of these are viral-like particle vaccines, and even plant-based vaccines. All right, so this is all I have for today about vaccines. I hope that was helpful in some way. Does anyone have any questions that they want to know the answer to? I can't guarantee I know the answer, but I can try. Oh, are you asking me whether or not I'd get vaccinated? Yes, of course. If anyone let me get vaccinated, I would 100% go get vaccinated. Any other questions? Yeah, some people are asking whether they should wait for a specific vaccine. I probably would try and get whatever was available at this point because it's a real challenge this is one of the only times where everyone on Earth has to get vaccinated within a fairly short period of time. Usually with the flu vaccine, it's flu season in different parts of the year at different times of the year. So we have a little bit more time to get all of those vaccines distributed to people. But all of the vaccines that are approved by the US are over 95% effective, which is good efficacy rates. They've all been demonstrated to be safe. So beyond that, I would just go for it. Any other general questions? Anything about viruses or vaccines or OSU Marion or antibodies? All right. Well, I'd like to thank everybody for coming to this session. I hope to see everybody at OSU Marion. You can come be in some of my classes and meet some of the other faculty members and take advantage of some of our great community and our resources and the other really awesome students. So thank you very much for coming.